Hello everyone, my name is Yahya and Naggar and I am a research associate at Faculty of Science Tanta University in Egypt. Currently I am Alexander von Humboldt postdoc at Hala University, and today I am going to talk with you about a topic that I think will touch all of you, and it's very important to think about it, which is pollinator decline and why bees are dying around the world and how we can protect them. So before we go ahead, let's have a look here. This is our food with bees, and here there is our food without bees. So it's very clear that bees make sense. So why bees matter? Well, bees are the most important pollinator of our plants, of our crops, vegetable, and third of the food we eat are dependent on pollinator. In addition to that, it provides us with a good source of honey, pollen, propolis, royal jelly, or even bee venom, and all of these products are of high nutritious and medical importance. So, what's wrong with bees? Well, over the last 15 years, there have been ongoing reports on science publication and on newspaper about the decline of honeybees and the consequences that might have. And unfortunately, there is no single factor have been identified but to explain this colony losses, and this syndrome have been called colony collapse disorder. Therefore, it's very important to identify or understand the driver behind this honeybee decline. And as I mentioned, there is no one single smoking gun. Instead, multiple causes, including biotic and abiotic environmental stressor, have been proposed. That includes the four B's, which is parasites, pathogen, poor nutrition, and pesticide. And nowadays, it's most acceptable that the interaction between all of these factors is the main cause of this colony decline. However, I am going to pay more attention about the exposure of bees to pesticide and pathogen and how they can interact with each other to affect the health of honeybees. Well, if we are going to talk about pesticides are chemicals that used to control the pest and disease of our crops, and it's still important to apply pesticide. However, the misuse of pesticide application causes the problem because honeybees during foraging, searching for nectar and pollen, are exposed to a lot of these chemical of different mode of action, and this chemical, besides the additives and the adjuvant that included in the pesticide formulation, might interact with each other and affect the health of honeybee and makes bees more susceptible to parasites and pathogen. Therefore, we are going to talk also about the pests and the pathogen of honeybees. Well, one of the most important ectoparasite of honeybees around the world is the varroa mite because it's distributed worldwide, and it transmits a cocktail of honeybee viruses during its feed on honeybees, hemolymph and fat body. And one of the most important virus that transmitted by varroa mite is the deformed wing virus that cause developmental deformities in honeybees and might lead to high level of overwintering colony losses. In addition to that, there is also another important honeybee virus, which is a black queen cell virus that have been detected in high rates in over or collapsing colonies. This virus attack the developing queen cell of honeybees and also of great importance. Another gut parasite, which is Nosema serrana, that reproduce in the gut of honeybees and cause nosomiasis disease. And as I mentioned, I would like to share with you the results of my recent experiment about the exposure to pesticide and the pathogen in honeybee. So I exposed bees to two newly registered pesticides, which is fluopyridifuran and sulfoxaflor. This pesticide regarded as the fourth generation of neonicotinoid because it shares the same mode of action like neonicotinoids. In the first experiment that have been carried out in United States before I came here to Germany, I exposed bees to the first pesticide, either the active ingredient or the formulated product, Sivanto, and I exposed bees during their larval stage when they are young or babies, and during early adult stage to very little doses of this pesticide and for a short time. Then later in life, I stressed them again with a gut parasite, which is Nosema serrana, to see how is pesticide exposure will affect the parasite load or not. And here is the result, which was very interesting, because we see that exposure to pesticide during larval treatment or adult treatment was able to increase the parasite intensity. However, this increase was depend on how strong or weak the colony, and this is very important point for beekeeper to select high resistant and strong colonies. In the second experiment, I exposed bees to this pesticide fluopyridifluran and sulfoxaflor under lab condition with another 3 viruses, the deformed wing virus A and B and black queen cell virus, either individually or in combination. And we checked viral load after 7 and 14 days, and fortunately we didn't find any synergistic effect, so the pesticide exposure didn't increase the viral load. At the end, I would like to share with you how we can protect bees or our pollinator. Well, we can support our local and organic farmers. We can start practicing organic gardening at home, planting bee-friendly wild flowers at our garden, providing some water sources during hot summer for bees, and most importantly, to say no to pesticide or regulate pesticide, and increasing the awareness about the importance of honeybees. And who knows, might be one of you will be a beekeeper and start his beekeeping business or industry in the future. And at the end, thank you so much, and I hope you enjoyed